All right, guys, here is the screencast for the Mendel and heredity information. And you had already taken Cornell notes using your textbook, but I just wanted to go over a few things, especially for the people in my 5th, 6th, and 7th who I did not see yesterday because of the snow day, and who I'm not going to see today because I'm at a meeting at the district office. So follow the instructions on the board, and hopefully you'll be okay with the monohybrid crosses. We'll do the dihybrid crosses. We'll review Friday. The test is definitely moved until next week. All right, this first part just talks about where our early ideas of genetics came from. And I would say early on, a lot of people didn't understand that genetics was actually based on dominant and recessive traits. They thought it was more kind of a blending of characteristics. So for an example, if you had kind of a shorter dad and a taller mom, you would kind of end up with an average height individual or child. And yes, this does hold true in some instances with some traits and characteristics, but a lot of times it's not a blending and we see the dominant and recessive. So Gregor Mendel is referred to as the father of genetics. And if we look at the dates in which he was living and working in the 1800s, 1822 to 1884, this is way before the discovery of the DNA double helix, which was in the 1950s. Our understanding of DNA, chromosomes, those terms and concepts didn't exist when Mendel was working with his ideas. So I think it's rather remarkable that without him knowing any of that information, he was able to see the inheritance patterns. He was a super meticulous data collector. He worked with pea plants in the monastery. He was a monk, so he lived in a monastery, and he basically laid the groundwork for modern genetics by breeding and studying thousands and thousands and thousands of pea plants. Couple terms that you want to make sure you have defined and know: genetics, the study of heredity and variation of inherited characteristics. Traits are those distinguishing characteristics that are passed from one generation to the next. And then one more, genome. That is, my genome is my genetic makeup with all of my DNA and genetic information. So you might be asking yourself, why peas? Why would he work with pea plants? Well, they were readily available in the garden at the monastery. They were rather cheap. They reproduced quickly. He was able to control the breeding, and I'll get to that in just a second. But basically, flowers have both male and female reproductive organs, so he was able to control whether or not he was letting them self-pollinate. So what that would be the male and female reproductive organs of one plant, or cross-pollinate using the male organs of one plant, let's say, and then the female organs of another plant, transferring the pollen there. So that was super important in his experiments because he knew exactly what he was breeding. He could use purebred plants, and purebred plants, whenever you see that word in reference to Mendel, you're talking about big T, either homozygous dominant or homozygous recessive. Purebred means you only have big T's to give or little t's to give, okay? Genetically uniform, that's what that says. And then also pea plants, one last thing, is they consisted of traits with only two alternative forms. It was basically an either or, so they were either tall or short. They either had purple flowers or white flowers. There was no in between, so that was also very important. Here's a little bit more on the pea plant's ability to self-pollinate. Mendel could interrupt this self-pollination by mating plants with a specific trait. So he started with purebred plants, removed the male parts of the flower, and fertilized the female parts with pollen from a different plant. So he was definitely able to control which plants with which traits he was crossing. On the next slide, there is a little video clip. Hopefully it plays and gets embedded correctly and you can hear it. It was a little bit small, but I played it on my computer and I recorded it on my iPad. But they're all from your textbook, the online resources on your textbook. But it basically just has a little cartoon Mendel going through in the garden, and it shows his experimental design.
Years, Mendel planted an estimated 28,000 carefully controlled crosses between different varieties of pea plants. He explored every possible combination, sometimes for up to 7 generations, and meticulously counted the results. One of the contrasting features that Mendel studied was the color of the plant's flower. One of his true breeding varieties always grew purple flowers, another always grew white ones. Their seeds formed the parent or pea generation. As soon as buds appeared on the vines, Mendel carefully opened them and removed the stamens to prevent self-fertilization. He later took pollen from purple flowers and sprinkled it on the pistils in white flowers. He also did the reverse. To prevent contamination by wild pollen, he then wrapped the flowers with protective bags. As a further control, he moved some of the plants into the insulated environment of a greenhouse. Weeks later, both groups of cross-fertilized plants developed seeds. Mendel stored the hybrid seeds over the winter and planted them the next spring. When the plants in this first hybrid or F1 generation grew flowers, they were all purple. Mendel called this trait the dominant one. He termed the trait that disappeared in the F1 generation recessive. Mendel let these F1 hybrid plants self-fertilize and produce new seeds. The next year, Mendel planted the F1 seeds, producing the next or F2 generation of plants. This time, there were some white flowers mixed in among the purples. He was well aware that traits sometimes skipped generations, so this was no surprise. But Mendel discovered something that no one else had ever noticed before. He counted the number of white and purple flowers. In similar fashion, he counted the results of crossing 6 other contrasting traits through to the F2 generation. When he worked out the ratio of dominant to recessive, in each case it came very close to 3 to 1.
All right, this picture, for some reason the picture didn't show up on this slide, but there was a picture in your textbook that showed the contrasting traits of the pea plants: height, tall or short, the position of the flowers, the color of the seed coats, the seed shape, the seed color, the pod shape, and the pod color. Now the important thing is to understand that, again, Mendel did all of these crosses. He recorded all of his data, and in every one of these traits that he used, he always got the same results. So this is showing his experimental design. He took a parent generation, all right, 2 purebred flowers. So let's use height, and I'll use T's. He basically crossed a big T, a tall plant, with a little t, a short plant, all right? When he did that in the F1 generation, he always got plants that were tall, or actually in this example, they're doing white and purple flowers. So we'll switch to that. But I'm not going to use P's. I'm still going to use T's because I don't like P's as a genetics letter. So all the resulting offspring had purple flowers, all right? And that's because if you look at this cross, the only allele that this parent can give is a big T. The only allele that this parent can give is a little t. So all your offspring are going to be big T, little t. That means all of them are going to have purple flowers, all right? Then in this F1 generation, what he did is he allowed them to self-pollinate. So basically you were taking a big T little t crossed with a big T little t. I'll do the Punnett Square for that one. Actually, I'll do both of these on the next slide so you can see it. But what happened again and again, time and time again on all of these things, in the F2 generation, that trait that seemed to disappear, the white flowers, remember in this generation, 100% of them had purple flowers. In the F2 generation, that trait reappeared in this generation. And I'll show that on the next slide using the Punnett Square. Okay, so that first cross was big T crossed with little t, little t. That was our parent generation. So what you do in a monohybrid cross, and I'll do another one of these at the end, this is our Punnett Square. We take the alleles of one parent and keep them together and either put them across the top or along the side. It doesn't matter. You're going to get the same results in the boxes. The important thing is just to keep that parent's combination of letters together, and then take this parent's and put them along the side. And then what you're showing in the boxes is the probability of what the genotypes of the offspring would be from a mating of these two parents. And again, here we have 100% of them being big T, or big T, little t. And in that case, it was purple flowers. All right, so that was our P generation. Then in our F1 generation, we crossed, or they were allowed to self-pollinate. So you're basically crossing two heterozygotes, big T, little t on one side, big T, little t over here. What your Punnett Square looks like, big T, big T, big T, little t, big T, little t, and little t, little t. This is that recessive trait, the white flowered pea plant reappearing in that F2 generation. And then all three of these are going to be your purple. Okay, and that is his 3-to-1 ratio. So he saw this over and over and over again in that F2 generation. The next slide is going to have a little video clip that kind of goes through the experimental design again.
With just his data and his imagination, Mendel came up with a model of how heredity works that remains the cornerstone of genetics. He concluded that life is not transmitted in one piece, as most people assumed, but in discrete particles, which he called factors, and which we now call genes. Using his particle model, Mendel was able to explain the great paradox of heredity, how both inheritance and variation can coexist, how like can beget both like and unlike. Mendel concluded that these invisible factors are passed along unchanged, which accounts for inheritance, but that sexual reproduction leads to new and different combinations of them, which accounts for variation. Mendel theorized that a trait is determined by not one, but two factors or genes. These factors also come in dominant or recessive forms. But Mendel made an important distinction between the nature of the trait, which we call its phenotype, and the nature of the underlying pair of genes, which we call its genotype. For his experiment with purple and white flowered plants, there were two possible phenotypes, dominant purple or recessive white, and three possible genotypes. Two dominant genes, in which case the phenotype is purple, two recessive genes, in which case the phenotype is white, and one of each, in which case the single dominant gene causes the phenotype to be purple. Mendel imagined that in the production of a sperm or an egg cell, the gene pair is split apart, with each gene ending up in a separate germ cell. Mendel called this process segregation. He also imagined that the direction of segregation is totally random, It can just as easily happen the other way around. So for any particular germ cell, which one of the gene pair it receives is totally up to chance, like the flip of a coin. This became Mendel's first law of genetics, the law of random segregation.
All right, and this is what I was referencing. The F2 generation always had a purple and white flower ratio of 3:1. Now, if you want to record your results in ratios, that is fine, but if you want to do them in percents, I usually prefer percents. It would be 75% to 25%. All right? The white flower trait didn't disappear. It had just been masked or hidden by those dominant alleles. Okay, so again, thousands and thousands of crosses to show this time and time again with all of those different traits. This is just a data chart that shows the traits that he was working with, the round and the wrinkled, so all of these would be your dominant traits. All of these in this column would be your recessive, and you can see that it's very, very, very close to a 3:1 ratio in all of these. So the conclusions, the traits are not inherited in discrete, or they are inherited in discrete units. It's not a blending. We know it's not a blending because in that F2 generation in Mendel's crosses, that recessive trait reappeared. And then make sure you are familiar with these. They were touched on in the little video clips. The law of segregation is just saying that organisms inherit two copies of each gene, one from each parent. And remember, those are carried on the homologous chromosomes. We said that the homologous chromosomes, one's coming from mom, one's coming from dad. And why this relates to meiosis is during meiosis, they separate. And remember, it's only one chromosome from each parent that's ending up in that haploid gamete. All right, so the egg cell is going to have 23. The sperm cell is also going to have 23. And that means there's one copy of each gene from each of those traits and characteristics. And then the law of independent assortment, what's happening basically in the sperm cell and how those segregate has nothing to do with the inheritance of a separate gene for any other trait. Now, what I want to do on this last slide is just do another example of a Punnett square, and I'll do the first one from the monohybrid problem set. Again, what you're going to do is you're just going to use that packet as your guide and for the questions. I'm not going to make you look on Moodle to find the questions. You have the questions, but I do want you to record your answers on page 198 and 199 in your interactive notebooks. Okay, so you can just number them. The first one, I believe, I don't have the sheet in front of me, but I believe it's big N crossed with big N, little n. And I believe that the big N is dominant smooth and the little n is constricted pods. So again, what you want to do is set up your Punnett square. Big N, keep that parent's alleles together. Doesn't matter top or side. Big N, big N, big N, big N, big N, little n, big N, little n. Now what you need to record is genotypic and phenotypic ratios. You do not need to write out genotype and phenotype every single time. Just do a G and a P. The genotypic ratio, remember, genotypes are the letters. So you want to write down all the different combinations of genotypes that appear in your offspring. And there's two. We have 2 that could be big N, so that's going to be 50%, and 2, or you can do a ratio, and 2 that are going to be big N, little n. That is also going to be then 50%. So now you have completed your genotypic ratio. If there was only one genotype, let's say they ended up all being big N, you just write 100% big N. And then for phenotype, remember, these are physically what they look like. So if you're looking at these, the homozygous dominants are obviously going to be smooth. The heterozygotes are also going to be smooth. So you're going to record 100% smooth for your phenotypic ratio. All right, then you're just gonna go through, if you could have that problem set done coming into class Thursday, that would be awesome. See you guys Thursday. I'm sorry I had to miss this afternoon for the meeting at the district office.